Hey everyone, can you guys hear me? Great. My name is Rene Brown. I'm a master's student here at the Kennedy Center. So with my project, I'm basically working with Chris. The first thing he spoke about, I'm basically working with him. So because we wanted something, a little bit different, I decided to present on this part of my research. So what I will be doing is just to compare decomposition rate of black needle rush, otherwise called juncus. I guess you guys know it better that way. And salt marsh, cord grass, leaf litter in impounded salt marsh. So just to give a little background of what salt marsh is, so these are like ecosystems that are typically characterized by low permeable surfaces, rich in organic peat, mud, clay, which sets a top of sandy tidal sediments. So these areas also tend to experience seasonal changes, and these usually create like dynamic environment for resident plant and animal communities. There are three main stressors that shape these ecosystems. So the first one is salinity fluctuation, and these are due to tidal inflows and freshwater inputs. The second would be erosion, and these are basically from water movement, and these can actually displace soil and plants. And the last one is an aerobic condition, and these are usually like saturated soil, limited, and this is usually limits oxygen availability for roots. So because of these stressors, organisms must adapt to survive and thrive in such challenging environments. Just the characteristics and like the role of salt marshes. So salt marshes are dynamic coastal ecosystem that lie interface of wetland and the sea. So constantly shaped by tide, salinity, and sediment movement. So these environments are incredibly productive and support a wide range of species. So from invertebrates, fish to migratory birds, creating which create essential habitat and food web and supports food web. So one of the most important processes here in organic matter is decomposition, which fluids, nutrient cycle, and store carbon, making it salt marsh vulnerable for natural climate changes. So they also provide ecosystem services like carbon sequestration, and they also capture and store atmospheric CO2. So as erosion control, these usually stabilize shoreline with dense vegetation. And finally, the plant that dominate these species are elophytes, um, especially adapted. So finally, the plants that dominate these systems are halophyte types, especially adapted to survive in high salinity. So the role of decomposition in salt marsh. Decomposition is central process in salt marsh ecosystem. It drives nutrient cycle by breaking down organic matter and releasing essential element like nitrogen and phosphorus, which supports plant growth and sustain food webs. This process also fluids coastal trophodynamics, linking detritus-based energy to higher trophic levels, like invertebrates and fish, as I mentioned before. So both biotic factors like microbial and fungal activity and abiotic factors such as temperature, salinity, oxygen availability. So directly, these usually influence the rate and efficiency of these compositions. So understanding these variables interaction is critical for predicting how resilient a wetland can be with like due to like disturbance and how the diversity is maintained under changing environmental conditions. So the two species that I'll be looking at is juncus and salt marsh cordgrass. And just an overview of these species. So juncus is found in mid to high marshes zone, and the key for erosion and is key for erosion and carbon sequestration. It tolerates salinity from 5 to 35 ppt and forms dense ribosomes network that provides stability and help it spread. Its structure and salt tolerance make it ecological vital and ideal focus for decomposition studies. And the next one is Spirobolus. These are also known as salt marsh cord grass. These are like fundamental species commonly found in low marsh zones. So these usually play like a crucial role by stabilizing sediments, reducing erosion, and providing foraging and nesting habitat for a wide range of wildlife, including bird, fish, and invertebrates. This species has a global distribution and demonstrate an impressive salinity tolerance from 3 to 94 ppt. This makes it high adaptable to variable coastal conditions. The structural difference between salt marsh cordgrass and other marshes plants, such as black needle rush, may influence how quickly their litter decompose, which turn impact nutrient cycle and ecosystem productivity. So I have two research objectives for this chapter, and the first one is to just compare the decomposition rate of black needle rush and salt marsh cordgrass leaf litter under varying environmental condition. And the second one is to just identify key environmental factors affecting decomposition in an impounded salt marsh. And my hypothesis are decomposition rates of black needle rush and salt marsh will vary due to indifferent structural features and chemical composition. And if you guys don't have an idea what these are, I have pictures and you'll see what I'm talking about. And the second one is inundated area will affect decomposition rate due to oxygen, temperature, salinity, and salinity changes. However, due to my bad, sorry. So my study area is basically the same place as Chris. We're both working at the same place. So assessment will be conducted on six brackish water impounded at Little Edisto Island in South Carolina. The site spans approximately 21 hectares of impaired salt marsh formerly utilized as shrimp aquaculture facility. So these have habitats like high marsh, low marsh, and open water, each offering distinct environmental characteristics. The impoundment differs significantly in terms of inundation regimen and geomorphic position, providing a gradient of ecological conditions. So my methods and how I approach these, I basically went out and I collected leaf litter samples. Over time, I dry them and then I created some mesh bag measured 30 by 30 centimeters, and I weighed 10 grams per species for each litter bags. I went out, I basically follow or plant survey transect to mark those GPS point. I had tomato stakes and I flagged them. So I had 7 bags per stake, 3 stakes per plant, and 2 transect per impoundment. If you do the math, total that up, it will be 252 total experimental units. 7 sampling intervals. So the first one was from 14 days to a year, 365 days. I also at each takes and each environmental interval that I went out to collect plant samples, also collected salinity, temperature, pH at each take per sampling date. So after all of that, when I'm back in the lab, so sampling and lab work, I basically wash the bags. So I would collect the bag, gently collect them, put them in like a Ziploc bag, put them in oil so decomposition would stop. So I would come back to the lab, wash off residue and stuff, like additional stuff. I would weigh the initial wet weight, record that, and then I'll oven dry at 70 degrees Celsius to a constant mass for a minimum of 72 hours. I would then record the initial dry weight of those samples. And after all that, I would grind those samples with a Willow grinder machine, and then I will ship those to main campus to have them analyze them for crude protein, hemocellulose, I can't, and cellulose using natural detergent fiber and acid detergent. So these are like after I collect the samples, I would then send them to main campus for them to analyze those chemical compoundments of the ground samples. So for data collection and statistical analysis, Solida bags will be collected and weighed at each sampling site. So wet to dry comparison after oven dry at 70 degrees Celsius for a minimum of 72 hours. So to analyze the data, calculated the decomposition rate of each plant species at each site. I will use a single exponential decay rate to model and leaf decomposition and calculate the decomposition rate. So I would also use ANOVA to compare decomposition rate between plant species. So for my anticipated result, plant samples at inundated area of the impoundment will decompose faster than samples at the drier location. So like dissolved oxygen, temperature, and salinity will affect decomposition rate. And my second one is that rate of decomposition will vary based on the structural and chemical composition of the species tested. And I also anticipated that Juncus will decompose slower than Spirobolus due to being lower in surface area and higher in total volume. So I haven't had much data so far, but what I came up with this. So this graph shows like the percentage mass remaining over time for both plant species across each impoundment. I found a significant difference between two plant species with a p-value less than 0.05. So overall, Spirobolus is decomposing faster as I predicted than Juncus, which indicates species-specific difference in decomposition rate. So within these wetland environment, it is also good because it could influence nutrient cycle rates in these marsh areas, which is definitely important for understanding overall ecosystem functioning. So this research will contribute to our understanding of salt marsh dynamics, especially on the various environmental stressors. The insight gained will guide effectiveness of wetland management and restoration efforts, helping practitioners design strategies that support ecosystem resilience. Ultimately, this work aims to enhance the long-term sustainability of salt marsh ecosystem, ensuring their continued function and benefit in coastal environments. So I just want to thank a few people that has always been there for me. So first, I would like to thank Dr. Anderson for always supporting my committee member, Dr. Thomas, and Dr. Amy, also Christopher for always going out and supporting me, Keegan and Andrew always for always being there for me when I needed them. And also James Kennedy for supporting my research. These are my sources. And if you guys have any questions, thank you.
Question relative to, I think you attributed decomposition rates to vegetative structure. Is that right?
Yeah.
And that's the only factor that you considered in terms of decomposition?
So far, yeah. I've had some data with like water parameters like the salinity deal, but it wasn't efficient or enough to really show it being, useful to it or doing anything or, like varies. So I didn't mention it because I haven't collected a lot as yet.
Right.
Yeah. So possible, onward going on more into my research. I'm pretty sure there will be more, but I just didn't have enough data pertaining to like other,
One thought, and this is just simply a thought, without any data to support it. The Spiralis community is more regularly inundated than the Juncus community. And I wonder if the alternate wetting and drying might contribute to the decomposition.
Yeah, that's pretty interesting.
So, yeah, and of course you're dealing with, as I understand, a semi-impounded situation. And so anyway, and then, but a Juncus marsh is known to be peat building with a lot of organic materials, and not flushed regularly. So anyway, those just some
Thoughts?
general thoughts, familiarity with the ecology of the plant.
Yeah, I thank you for that. Any more questions?